So today we will continue to talk about herpes viruses in general and some of those types in particular. And we're going to start with a conversation about cytomegalovirus, which is beta herpes virus. And this virus is usually transmitted by bodily fluids, early in life, usually in the childhood. It's a common disease transmitted in the daycare centers. The primary infection is asymptomatic, or sometimes cytomegalovirus can cause something like common cold or mononucleosis-like illness. This virus, upon entry into the human body, infects monocytes, white blood cells, and becomes latent in the monocytes. It can stay latent in the immunocompetent individual all throughout life without causing any symptoms as far as we know. However, in the people who are immunocompromised, like people with AIDS or transplant patients, cytomegalovirus can cause pretty severe infections, ranging from infections of the eye, like retinitis and keratitis, to acute gastroenteritis, liver disease, and in severe cases, meningitis. Cytomegalovirus is the leading cause of the failure of a kidney transplant. It turns out that in patients who receive kidney transplant, they undergo chemotherapy. They undergo immunosuppression. If the donor is cytomegalovirus positive, then this virus will be transferred to a recipient of the kidney with a transplant. Upon the chemotherapeutic immunosuppression, the patient becomes immunocompromised, virus reactivates in the transplanted kidney and infects epithelial cells of the renal artery. Upon the infection of epithelial cells, response to the infection is inflammation. Inflammation results in repair. Repair of the renal artery results with the stenosis of the renal artery, narrowing, lack of proper blood flow, hypoxia, and kidney death. So you see, in this case, failure of the transplant is not due to the immune response. It is due to the proper lack of proper blood flow. Cytomegalovirus causes extremely severe congenital infection. This table here summarizes, you don't need to know the numbers as ever, it summarizes the sequelae of the congenital cytomegalovirus infection. So you see, first of all, the majority of infections are symptomatic, meaning that kids do develop symptoms of cytomegalovirus infection. The leading complication of congenital CMV is hearing loss. Many children develop cognitive deficits. Certain percentage develop motor deficits and seizures. So, unfortunately for newborn patients with congenital cytomegalovirus, the only available treatment are different inhibitors of DNA synthesis, like mirabegir, ganciclovir and acyclovir, and the new experimental drug called mirabegir. Now, you can imagine that newborns can tolerate the drugs much worse than adults, right? So, in this image, you can see a newborn with a congenital cytomegalovirus infection. This girl passed away 6 days after the birth due to the complications. these spots, this girl's skin, are actually sites of myeloproliferation, sites of the extramedullary hematopoiesis. So basically what happened is myeloid progenitor stem cells migrated out of the red bone marrow, ended up in the skin and started to produce blood. It's just one of the many, problems associated with the congenital CMV. This is why pregnant women are tested for the cytomegalovirus infection during the pregnancy, and at certain points the precaution, like if it's acute cytomegalovirus infection and it is early on during the pregnancy, often doctors may recommend to terminate the pregnancy because risks outweigh any possible positive benign outcomes. Interestingly enough, cytomegalovirus is associated with a cardiovascular disease. Now, association does not mean causation, as we all know. Now, cytomegalovirus has been linked to the development of atherosclerosis, high blood pressure, increased risk of coronary artery disease. And I had the privilege to work on the synergy between obesity and cytomegalovirus in mice for 5 years. And our group also demonstrated that cytomegalovirus worsens cardiovascular symptoms caused by hypercholesterolemia in mice. So mice had super high cholesterol. If you put that together with cytomegalovirus, it gets worse. So there are studies in mice showing that cytomegalovirus looks like it's promoting hypertension in mice, but again, the problem is it's all done in mice. In humans, studies are association studies. So we look at the number of people, at the people who are seropositive, who had cytomegalovirus infection, and then we'll look at the frequency of atherosclerosis in that particular group of people. Is there any other evidence? Well, the brief answer is yes. CMV can be found, this virus can be found in the atherosclerotic plaques inside of them, which is not really surprising because there are white blood cells in the atherosclerotic plaques. So monocytes will stick to atherosclerotic plaques, bringing virus in there. I am personally quite sort of, okay, what if cytomegalovirus does promote development of atherosclerosis? Look at this table. Not everything, of course, but I'll make some highlights. So this is a seroprevalence, meaning how many people in a certain cohort were infected at some point of their lives with cytomegalovirus. And remember, this virus remains latent, so once you're infected, you have it. Okay? So we're talking about 70%, 50%, 60%, 60%, 90%. I'm not nitpicking. There are some cohorts where prevalence is kind of low, but general idea that by the age of, say, 60, the seroprevalence in the population can reach stably 60, 70, sometimes 90%. Now think about this. If we have, let's do conservative estimation, if we have 70% of population infected with cytomegalovirus, it's not a risk factor anymore. It's the background. Does that make sense? Do you understand what I'm saying? It's a background. If you have 1,000 people who all smoke, you can't say, oh, like the entire population smokes. Does smoking cause lung cancer? Yes, it does. But in this case, it's kind of intrinsic risk factor. You know what I'm talking about? Like that's the lifestyle. Okay? We can say, if everybody would be eating absolutely terrible diet and everybody would be obese, obesity wouldn't be a risk factor for cardiovascular disease. It would be just the way of life for the entire humanity. So when we have a population, like 70% of population positive for CMV, I mean, when you have a patient, you kind of better presume that this patient is CMV positive. It's safer. Does that make sense? You know what I'm trying to say? Now, slightly different situation is with the brain. So, CMV is implicated into what's called oncomodulation. Doesn't mean that it causes cancer. It means that it can promote cancer. CMV has been associated with a cancer called glioblastoma. Now, as you can imagine, it's much harder for viruses to cross blood-brain barrier and end up in the brain. So that may be a risk factor here. Okay. The third group of viruses, herpes viruses, are gamma herpes viruses. And we're going to talk about two here, Epstein-Barr virus and Kaposi sarcoma virus. So Epstein-Barr virus was discovered in Africa. There's regions in Africa where it's the most prevalent. It was discovered in Africa by American physician, last name Epstein, and French physician with last name Barr. She's a woman. So they were studying the unusually high rate of nasopharyngeal carcinoma in the African population and eventually managed to link nasopharyngeal carcinoma to the virus that was later called Epstein-Barr virus. This virus, when it is acquired, it causes very well-known disease, infectious mononucleosis. Okay, people develop lymphadenopathy, loss of appetite, fatigue, high fever, sore throat, swelling of tonsils. That partially comes due to the fact that Epstein-Barr virus can infect epithelial cells. That's going to be tonsillitis, cough, fever. And it also infects B cells. That will contribute to lymphadenopathy and splenomegaly. So once infectious mononucleosis is over, virus remains latent in the B cells. And then from B cells, it can, in case of reactivation, spread back to epithelial cells. And in some cases, as we just learned about Africa, it can cause a cancer derived from nasopharyngeal epithelium, nasopharyngeal carcinoma. A latency in B cells contributes to the ability of this virus to cause lymphoproliferative disease known as Burkitt's lymphoma. These are two cancers directly caused by EBV. Interestingly enough, Epstein-Barr virus has been implicated in a variety of head and neck cancers, but not by itself. Epstein-Barr virus plus papillomavirus is kind of a new found recipe for high risk of head and neck cancers, throat cancers, nasopharyngeal cancers, all kinds of diseases. Now, there is a disease that you may or may not have heard about, chronic fatigue syndrome. Have you heard about chronic fatigue syndrome? It's a very interesting and unfortunate disease. It's usually diagnosed by exclusion. So people with chronic fatigue syndrome get tested for all known genetic, environmental, toxic, infectious conditions. When all other culprits are eliminated, they diagnosed with chronic fatigue syndrome. The main symptom is chronic fatigue. You have to appreciate it's not fatigue in the sense of you were raking leaves or mowing lawn or working in the yard 12 hours during the summer day, and you overexerted and you're just too very tired. People want to go and grab a glass of water, but they can't. they have no energy, meaning like they have extremely low levels of ATP. They cannot move. It is debilitating disease. It's a chronic disease. It lasts for years. There is no cure, and there are very futile treatments. Treatments mostly involve cognitive therapy, which is marginally effective, and some physical therapy, some exercise, which, as you can imagine, is also marginally effective because those people have a hard time moving. So forcing them to move doesn't make them much better or much happier. So Epstein-Barr virus was implicated in the chronic fatigue syndrome, but you can imagine that we talked about Koch's postulates. Ideally, to fulfill Koch's postulates, we need to collect more or less homogeneous population of people at a certain age. We're talking about thousands and thousands of folks, infect them with Epstein-Barr virus, and then have a control group that will not get EBV guaranteed. It's impossible, logistically and ethically, to do experiment like that. Okay, so we have to do observational studies, and epidemiology is fine with that. We're going to be doing observational studies. We're going to be doing sequencing. We're going to be doing, identification of antibodies and all that kind of stuff. And maybe eventually we will be able to prove link or disprove the link. Well, science doesn't really disprove anything, just finds no link. Okay. Now, another virus that belongs to that same group, Kaposi sarcoma, is a skin cancer. So this virus infects connective tissue, dermis of the skin, hence sarcoma. It's one of the hallmark diseases, the AIDS patients. It's awkwardly common in Mediterranean countries like Italy, North Africa, South Spain. Now, when I say, and we're going to run into this every so often, when I say hallmark disease, AIDS patients, so you have to distinguish patients with HIV, patients with AIDS. Patient with HIV is just a folk who was infected, who acquired HIV, may have no symptoms whatsoever except maybe for a low T cell count. Patient with AIDS have very progressed immunodeficiency, and patient has all kinds of symptoms associated with that immunodeficiency. So what you can expect is cough, either from tuberculosis or fungal pneumonia, diarrhea from various protozoan infestations, complaints about diarrhea, this purple spots on the skin due to the Kaposi sarcoma, often anal cancer due to the papillomavirus infection, in some cases, in many cases, fungal meningitis, characterized by confusion, headaches, and stiff neck. Now, this kind of, if you see a patient who comes with like 2, 3, 4 of those symptoms, it's maybe not a bad idea to address specifically T cell counts to see if the patient has AIDS and then, move accordingly. But the problem with this disease, it's a cancer. And it's a skin cancer. You cannot surgically remove the skin. So patients with Kaposi sarcoma, they pretty well advanced in the AIDS, so it's futile to treat it because it's cancer, and chemotherapy has limited efficiency. Does that make sense? Any questions about herpes viruses? Okay. Yes.
So it's not prejudice, just evolves from the AIDS, like it's a result of being a progressive from the AIDS.
Say it again.
It's not something that you catch. It's something more so. It's just as it progresses, it turns into it.
Which one are you talking about? Kaposi sarcoma?
Yes.
So Kaposi sarcoma, that's the different virus. It's a herpes virus, okay, which you may catch as a child or as a adolescent or adult. And then it's going to be latent in, I believe, in B cells, some immune cells in the skin. So it stays there. Then, when you, God forbid, catch HIV and develop AIDS, this virus gets reactivated. And this virus causes cancer on the background of limited immunity because of the HIV infection. Does that make sense?
Yes.
Good. Sure. So we move on to talk about DNA replication. Now, one of the reasons why we pay so much attention to basic mechanisms of gene expression and DNA replication is because some of those mechanisms are unique to bacteria, and they are quite different in bacteria compared to eukaryotes, which gives us a great opportunity to selectively inhibit, say, bacterial protein synthesis without affecting eukaryotic protein synthesis, or inhibit bacterial DNA replication without affecting eukaryotic. Does that make sense? So this fundamental studies actually gave us a whole bunch of antibiotics in the end. So let's talk about DNA replication. So every time cell divides, DNA is replicated. So you have to understand that if cell isn't going to divide, it is not going to replicate its DNA, because it's going to be a waste of time and resources. Make sense? So DNA replication is basically a prerequisite for the cell division. If DNA is copied, cell will divide pretty soon. Okay. The main purpose, of course, is to pass the genetic information to the next generation. So how does that work? This is an example of DNA replication in eukaryotes, which is similar to prokaryotes. So double helix is unzipped. It's so-called origin of replication. Origin of replication is reached in the AT pairs. Why? What is the importance of it being reached in AT pairs?
Because they have the fewer amount of bonds.
Yes. So they have fewer amount of bonds, meaning they
Can break apart easily.
they are easier to separate. Okay. So at the origin of replication, DNA forms two replication forks. So those are replication forks, which importantly move in opposite directions. Okay? Does that make sense to you? So think of it. I don't have zipper like this here to show you, but you know the zippers with two running things? I don't know how this running things, like the actual dongle that you move up and down, how it is called. But you know the zippers? So imagine these two dongles are in the center, and you start to split them to the sides. So each dongle will represent an individual replication fork. Okay? Now, usually bacterial DNA has one origin of replication. We can count as one origin of replication in bacterial DNA. Remember, bacterial DNA is circular. So if this is origin of replication, when it starts, what you're going to see is pretty much this. So that's one replication fork. This is another. Okay, that's very schematic. Okay. Now, bacterial DNA is somewhat small. Okay, eukaryotic genomes are usually much larger. For that exact reason, eukaryotic DNA usually has multiple origins of replication, as shown here. And these multiple origins make replication pretty fast. Okay? That makes sense? Now, it's not just one enzyme that participates in the replication, it's actually a whole set of enzymes with a different function. The main rule of replication is that DNA is copied under the complementarity rule. So if there is A, there's going to be T in the new strand across this A. There's G, there's going to be C across it in the new strand. All right? So far we're good? Now, I want to make a pitch for scientific method here, telling you a shortened story of the study of DNA replication. So scientists, I'm not going to say that I'm terrible with names, when they were figuring out the mechanism of replication, they made 3 equally possible hypotheses. First hypothesis, and that's how science works, that's how you do it. One hypothesis was that we have a DNA molecule, double-stranded DNA molecule on the left. You see that? Okay? And then this double-stranded DNA molecule, after one round of replication, it produces, you're going to have 2, right? It's replication, it's copying. So one hypothesis stated that these 2 molecules is going to be the original one and another molecule that consists of 2 completely new strands of DNA. Okay, so 2 old strands in one, 2 new strands in another. Does that make sense? Second hypothesis was different. They said, okay, so we have the original one. After one round of replication, we're going to have 2 molecules. Each molecule consists of 1 old and 1 new DNA strand. There was a third hypothesis, which for the sake of brevity I will skip, okay? So basically, they called these 2 strategies of replication, they called left one conservative replication, and they called right one semi-conservative. Okay, so conservative meaning that the original molecule is completely preserved, semi-conservative that, partially preserved. And they did an experiment. And you know what? It turned out that only one of the hypotheses was correct, and correct hypothesis was this. DNA replication is semi-conservative, period. But why I use it as an example for, the scientific method. When scientist formulates the hypothesis, it isn't a statement that must be correct. It's a hypothesis that can be proven wrong. The really good saying, really the sign of discovery is not eureka. The sign of discovery is the phrase, it's funny. When something unexpected happens, then it's a discovery, then it's interesting. When you get the result that you expect, it's kind of boring. I mean, it's rewarding, but kind of boring, right? So, in this situation, these fellows had 3 hypotheses. Only one of them was correct. That's it. It works, okay? Now, as we discussed, each strand of double-stranded DNA, each strand carries the entire compendium of genetic information about an organism. So obviously, second strand works as a backup. If there is a damage or a mistake or something else in one of the strands, second strand can be used as backup to restore the integrity of the molecule. We have to highlight that there is a certain difference between the replication in the bacteria and eukaryotes. In bacteria, as you remember, DNA is not separated from the cytoplasm. So technically speaking, presence of DNA in the cytoplasm is kind of given, okay, normal, right? In eukaryotes, DNA is in the nucleus, therefore Replication is in the nucleus. So DNA, either double-stranded or single-stranded, should not be present in the cytoplasm. And actually, in many eukaryotic cells, there are enzymatic receptor systems that detect DNA in the cytoplasm and activate cell death, because that's not normal. In humans, the system carries a very funny, in my opinion, name. It's an acronym, and the acronym is STING. I don't remember what it stands for, but like a set of proteins called STING. Any questions before we move forward? Now we're going to spend a lot of time here. And the deal is, I'm going to go step by step, very slowly. And I don't want to move forward until, everybody kind of nods or blinks once for, yeah, we got it. Okay? So we're going to start over here with an enzyme called topoisomerase or gyrase. Now, what is gyro?
Top.
No. What is gyro? Like, did you ever had gyro? Food, yeah. It's not sandwich. It's kind of wrap, right? It's kind of, that's important. It's wrapped. It's like, so DNA, double-stranded DNA molecule, isn't straight. It's all wrapped. Okay? Does that make sense? It's all wrapped. Basically, my favorite, and it's pretty accurate analogy, have you ever dealt with this old-style phones where the phone is on the long helical wire? What happens to that wire all the time? Twisted and tangled. So that's what happens to DNA molecule. The function of topoisomerase or gyrase is to untangle the DNA, straighten it up. That does require ATP. Once the DNA, okay, we got it, this function, so gyrase untangles it. It's called DNA supercoiling. So topoisomerase 2 or gyrase, it prevents the supercoiling of the DNA. Next, you need to unzip the DNA molecule. And this is the function of the enzyme called helicase. Right here. Okay? This blue triangle is the helicase. It unzips DNA molecules. Now you can imagine that it's kind of a forceful process to unzip. It also requires energy, requires ATP. But not only this, those two strands, they, due to the energy requirement, sort of, minimum energy of the system, these strands try to get back together. Does that make sense? Okay, do you understand what I'm trying to say? They are pretty well connected by hydrogen bonds and they're much more stable being together. In order to prevent these strands to collapse back into a double-stranded DNA, single-strand binding proteins, or SSBP, shown here, they bind to these strands, and these single-stranded binding proteins will be like a slide going over this unzipped DNA molecule. You can take, yes?
Where do they bind? To the nucleotide, or are they...
That's a great question. They big,
Okay.
so they will bind to each individual
Like the whole
chain.
shape?
They most likely they have a fragment that is called a zinc finger. It's kind of a weird name. Zinc finger is the common DNA binding fragment in a whole bunch of enzymes. It looks like a finger. And there's a pocket to bind DNA molecule, and they will prevent those chains to come back together. Before we move any further, we're clear so far. So we got it untangled, no supercoiling. We got it unzipped, and we stabilized this replication fork right here using single-stranded binding proteins. Okay, I'm gonna remove that stuff. So what happens next? Now, if you look, when you look at the image here, which is a pretty good image, you can see that this image is a blow-up of that particular rectangle, right? Do you see that? So it's kind of a, okay, again, look at this one. You see the rectangle over here? This is what you see at, what you look at in this large image, okay? Does that make sense? Here's the problem. Replication is the dynamic process. Things are moving. What you look at here is a snapshot. Does that make sense? Like you look at the photograph of people running, you didn't know how was the start and what's going to happen at the finish. You see one particular moment of the race. Make sense? So this is why we'll have to refer to certain things that aren't on this large picture. So once the DNA gets unzipped at the origin of replication, the first thing that happens is the synthesis of so-called primers. I don't know if you can see it, that primer, that specific primer is incredibly small. It is right here. Tiny little green dot. It's basically the same as this primer, structurally. So what are primers? Primers are short fragments of RNA that are required to start the replication. The function of the primer is to set the starting position for the DNA polymerase. Am I clear? So you cannot replicate DNA without a primer. Okay, so we have RNA primer. And I'm going to be talking, for now, I'm going to be talking about this upper, top strand. Okay? Does that make sense? We're going to give it a name in a second. Does that make sense? So we've got a primer. Everybody with me. We're all good? Everybody's here? Say it one more time. From the function of the primer. Primer marks the starting point of the replication for the enzyme that actually makes a new strand of DNA. It's like a tag saying start here. And just as the tag, it will eventually be removed. So far, you with me? Okay. So the enzyme called DNA polymerase 3 binds to that site. So in the very beginning, that DNA polymerase 3 was right here. Okay? Right here. And it moves to the left. Does that make sense? No? Give me a second. DNA molecule. Original DNA molecule, okay? Primer. You with me? DNA polymerase 3. You with me? DNA polymerase 3, in order to start, it has to bind to that complex of primer and DNA. You following so far? Good? Once it's bound, it can start moving. And it will insert nucleotides. It will add them to the primer and then forward, like this. And it will add them in a complementary manner. Does that make sense? Let's say CTAGGCAT. What you're going to get in the new strand is GATCCGTA. Does that make sense? And they will be inserted one after another, one after another. Does that make sense? Now, we're coming to a very important law of replication. DNA polymerase 3, enzyme that actually builds a new strand, will always read the original DNA, which is blue, like that's original, from 3' to 5'. No exceptions. Does that make sense? Are we clear? Always, absolutely, always, 3 to 5'. Now, my next question for you is this replication fork. And I want you to use the terms like left and right. It's totally fine with me. On this slide, this replication fork, which direction does it move? Left or right? That's a simple question. Left. It started over here, and it moves this way. That particular replication fork, this one, it moves to the left. Everybody's fine with that statement? Now, the direction of the top strand, top original strand, 3 to 5' goes which? So 3' end of the top strand is where? On the right. 3' end is on the right of the top strand. Does that make sense? Original one, the blue one. Got it? 5' is on the left. I told you before, if I'm going to ask you a hard question, I will announce that it's a hard question. I want you to kind of, doing it bit by bit. So the fork moves to the left, and the DNA polymerase 3, this one, will be reading the blue strand, this one, this top blue strand, in which direction? From right to left. We're clear. So it's going to be uninterrupted synthesis. The fork moves to the left, DNA polymerase moves to the left, everybody's happy. Does that make sense? The strand that is synthesized in the uninterrupted fashion is called a leading strand of DNA. Yes?
So the fork that keeps moving to the left keeps unzipping and uncoiling the
Yes.
strand
Yes.
continuously
Yes, absolutely. That's really good note.
until it reaches the stop
We'll-
codon?
We'll get to this. No. No, we'll get to this. Hold on a second. We're going to talk about only one fork at a time. Okay? Does that make sense? 4 goes to the left, DNA polymerase 3 goes with the fork. Perfect. Right? That top strand, the leading strand, will be produced in uninterrupted fashion. Cool? You have another strand at the bottom. You see this one? You have this strand. Can you just put, look, that's another strand that you can copy, right? And you will, but the problem is, where is 3' end? On the left, 5' end, on the right. So DNA polymerase 3, can it follow the fork? No. Everybody got this? So instead, DNA polymerase 3 does jumping. So let me show you what happens here. This right here is a primer. Can you see it? And this is a primer. Okay, so these are two primers that are positioned fairly close to each other. And each primer sets the starting point for DNA polymerase 3. Before we move on, are you comfortable with that statement? We have two starting points for DNA polymerase 3. Now, if you would look here, you will see that DNA polymerase 3 synthesized a short DNA fragment. So it used to be here. Now, this DNA polymerase 3 kind of jumped back over here, and it synthesizes another fragment. This fragment that is being synthesized right here, where is it going to end? That's a simple question. This fragment that is synthesized, where is it going to end? I mean, where is it going to stop? Right there at the primer, right? That's it. DNA polymerase 3 then will jump back onto the next primer. We'll make another short fragment, and it will keep jumping like a grasshopper. Does that make sense?
Logically, no. But
It's not about logic, because
I'm just saying.
No.
It doesn't make sense.
here's the deal. So DNA, well, I can try to make a sort of a plug for logic. Think about this. DNA polymerase 3 is essentially a reading enzyme. If you set the rule that reading from left to right, say you're a teacher, you have a classroom full of kids, and they all know that they should read left to right, and then you have one renegade who decides that it doesn't matter. And the kid starts to read right to left. What comes out is gibberish. So DNA polymerase 3 can only read in one direction. You cannot not replicate the second strand, because then you're going to have a mess of a single-stranded DNA, a double-stranded DNA, and a single-stranded, like loose strand just dangling in there. Does that make sense? So the only way to copy that strand that is in reverse is for DNA polymerase to make it into short fragments. Think of it maybe this way. You have a book, and normally, and say somebody told you, you have to copy that book. Normal person would copy from chapter 1 to chapter 2. Imagine you're put in a position when you are given chapter 13, then chapter 12, then chapter 11, then chapter 10. Does that make sense? So you're kind of going backwards, but you're not copying backwards. You copy chapter number 13, and then you jump back to the beginning of chapter 12. Copy chapter 12 and jump back on the beginning of chapter 11. Does that make sense? So you do these jumps. I mean, I used to do almost like a circus here, when one person was walking straight, representing leading strand, and I was like jumping back and then moving forward. It required a lot of physical exertion on my side. But basically, what you end up with on that second strand over here is discontinuous replication. Discontinuous, not continuous. Those fragments of DNA that are synthesized are called Okazaki fragments. So before we move on, let's sum up what we've got. I'm going to skip the unzipping and, untangling, unzipping part. So we have two strands. Leading strand is produced continuously because 3 to 5 prime direction matches the direction of replication fork movement. Lagging strand here, synthesized in discontinuous matter, because 3 to 5 prime direction is opposite to the direction of replication fork movement. And lagging strand is composed of Okazaki fragments. It's just named after the guy who discovered this replication feature. We have two problems on our hands. Problem number one, primers. What are primers made of?
RNA,
RNA. Which molecule are we synthesizing?
DNA.
DNA. So we got to replace RNA with the DNA. Correct? The function to replace RNA with the DNA is laid upon the enzyme here that is called DNA polymerase 1. Okay? DNA polymerase 1 destroys the RNA primer and fills the gap with the DNA. Am I clear? Good?
It destroys.
RNA primer removes it and fills the gap with the DNA. I can't find really an analogy. Okay. So far, does that make sense? Problem number two. It comes from the chemistry of the enzymes. Question? Lena?
No, I know, we got it.
Okay, we got it? Does that make sense? So all primers, it's not like, the whole system is waiting for the replication to finish, and then primers are removed. No, they remove dynamically, sort of, when they're not needed anymore, they destroyed. We're clear. So you see it here. Primers are removed. Now, problem number two, chemically, it turns out, we kind of have to take it for granted. Turns out with DNA polymerase 1 replaces the primer, it cannot connect the ends over here. Does that make sense? So these ends of two adjacent Okazaki fragments have to be stitched together. So you see these ends not merged. These ends of Okazaki fragments have to be stitched together by the enzyme called DNA ligase. Okay, this enzyme literally stitching DNA fragment into a strand. So far, does that make sense? So what you need to know, in my opinion, best way to learn this thing is to describe it to somebody else. So if you have any relative who is physically inferior to you or depends on you in one or another way, tie this person to a chair and start explaining the DNA replication. You need to know the functions of the enzyme, so I'm going to ask this. You need to know the difference between lagging and leading strand. Little digression about the naming. Doesn't it look counterintuitive that DNA polymerase 3 is the main honcho here, and DNA polymerase 1 is merely a sidekick replacing the primers? Well, the enzymes were named in order they were discovered, not in order of importance.
Is there ever a second one?
Yeah, I think so. I don't think it's like critically important, but there's DNA polymerase 2. So that is always weird. Okay. Now, since we're focusing on bacterial replication, I'm going to answer Lisa's question now. What happens, when does it stop? So a few things. Now, we're going to enter another pretty interesting concept here. Look at the image, small image, on the right-hand side. How many replication forks do you have on the right-hand side then? Here. How many replication forks? It's a simple question. How many forks? 2. One goes on the left, to the left, one goes to the right. Does that make sense? Which means that in the fork that goes to the left, the top strand will be leading. Does that make sense? But in the fork that goes to the right, the bottom strand will be leading, because 3 to 5 prime direction flips. Does that make sense to you? Can you see that? Kind of picture it. You understand what I'm saying? No? Okay, again. Leading strand is the one in which 3 to 5 prime direction matches the direction of replication fork movement. It's a long statement, but let's see. Replication, so we're going to call it left replication fork. Left replication fork. Where does it move? Left or right? Left. Top strand. The direction of 3 to 5 prime is what? To the 3 to 5 prime. To the left. So top strand, 3 to 5 prime to the left. Left replication fork moves to the left. So in this part, top strand will be leading. Let's take a look at the right replication fork. It moves which direction? To the right. Look at the bottom strand. 3 to 5 prime goes to the right as well. Does that make sense? So leading strand and lagging strand isn't something intrinsically wired into a DNA. Makes sense? We're getting closer to Lisa's question. Look, I'm going to draw it in the empty space. As good as I can do. So that's circular, double-stranded DNA chromosome. Does that make sense? You with me? These are 2 forks. Okay? That makes sense? Now these forks are moving. I'm kind of trying to draw a little kind of moving cartoon here. These forks are moving. This fork on the left moves which way? It's going to be what? Counterclockwise, and the right one will move clockwise. You with me? So I'm going to picture the next step. So imagine these forks are all the way here. So I have this fork, you have DNA molecule synthesized, DNA molecule. Those are rather new strands. Does that make sense? What's going to happen to these forks eventually? Keep moving them. They're going to do what? Then you run into each other, right? And that's it. That's a stop. So these forks, as they move, they're going to run into each other and they will stop. Does that make sense to you? The only issue that bacteria face with that process is that they do have 2 double-stranded chromosomes after the cell division, but these chromosomes are concatenated. It's a fancy word, concatenated, which means, let me try and draw it as good as. So this is one circular chromosome. I'm not drawing 2 strands, but you get it just. And this is another. Does that make sense? Do you see that? So they are like 2 rings, like this, 2 rings of a chain. Does that make sense? So how do you separate them? An enzyme called Topoisomerase 4 does this. That's it. Breaks, nicks one chromosome, separates the ends, closes them after. Yes.
So since you're showing us circular,
Yeah,
is this bacterial or is
that's
this
bacterial.
centrifugal?
That's bacterial. In eukaryotes, there's a huge problem. I'm going to address it really quickly. So imagine now, before we move on, like this is what I'm going to ask you on the exam. Okay, like this, topoisomerase 4 separates concatenated strands. That's it for the exam. Does that make sense? What we're going to talk about, like briefly mention eukaryotes, not on the exam. Any questions about bacterial replication? Okay. Here's the problem. Look at this one. Imagine for a second on the left, like that black square that I drew here. Imagine for a second that this is eukaryotic DNA. It's linear, right? So the leading strand is it going to copy all the way down to the end? That's a simple question, no tricks. Sure. Yeah. I mean, it can go all the way to the end. The lagging strand, there's a problem. You got to put a primer over here. Turns out DNA polymerase cannot replace that initial primer. So lagging strand will lose a bit of nucleotides when DNA is replicated. And there's an enzyme called topoisomerase that extends that end. So eukaryotic chromosomes have fragments on the ends called telomeres. Basically, it's like a disposable DNA, largely disposable DNA, that is supposed to get shortened every time cell divides. It's kind of, they're supposed to lose some, nucleotides and stuff. Make sense? At some point, telomeres become so short that cell dies. It's normal. So, in humans, it's kind of a double-edged sword. Technically we know that if we can stimulate the activity of telomerase, the enzyme that keeps telomeres long, we can make cells to live forever. Is that a key to immortal life? Well, I don't know about immortal life, but that looks like a key to cancer. Because in cancer cells, telomerase is abnormally active. It keeps those ends long. Cells survive way more than they're supposed to survive. Accumulate mutations, may become malignant, and eventually it leads to cancer. So, it's a pretty cool story. At some point, telomerases were like a hype of molecular biology, and folks who discovered them, they got Nobel Prize. They deserved it. Now it's kind of slowed down. Okay. We're good? Now, this table summarizes. I can tell you from the get-go, one thing I'm not going to, there's no question about it on the enzyme. I'm going to give it away. Not going to ask you about the sliding clamp. Not much relief here. It's just one of what, like 10 or something? But sliding clamp is kind of boring. Okay? Are we good? Any questions on replication? Mutations. Few things. Mutations are changes in the nucleotide sequence of DNA or RNA. There is an expression that even scientists use sometimes. The expression is mutant protein. It's technically incorrect. Protein is not mutant. DNA can be mutant. RNA can be mutant. Protein can be altered, dysfunctional, changed, but not mutant. We're clear? Now, Yes. So, mutations can occur exclusively in the DNA or RNA, but, okay, RNA. the statement mutant protein or mutations in the protein is technically incorrect? Well, because it's not where mutations occur. Right? We're good. Awesome. That's number one. Number two, where can they come from? Source number one is synthesis. So let's talk about RNA synthesis first to get it out of the way. Normally in the cell, RNA is synthesized in the process of what? Transcription. Speak up. Look, if you say something that is incorrect, nobody's going to embarrass you. Nobody's going to make fun of you. We'll just say, okay, that's wrong, and we'll move on. Okay, so please don't hesitate to say something that you're not sure about. It's totally fine. Transcription mistakes. In which process in the cell a DNA molecule is synthesized?
DNA replication.
Replication mistakes, then. What is the role of RNA in the cell, generally speaking, like mRNA? To transfer message, to synthesize proteins, right? It's a messenger RNA. Do we really care about mistakes in the message? Do we really care about the mistakes in our scribbled recipe?
No.
We don't really care, because if that chocolate cake comes out as the baked chicken, it's not a big deal, you know? That makes sense? We can make it again, right? So RNA synthesis is very unreliable. When RNA is synthesized, there are a ton of mistakes, which actually explains why RNA viruses so easily mutate. It's kind of a bit of a digression, but who got the flu vaccine this year? I'm not pleased. It's a little coverage. Should be more. Well, I got it too. Some of you may have flu this year, because flu mutates really fast. And you will need to take another vaccine next year, because it mutates. It's RNA virus. When its RNA gets copied, there are plenty of mistakes. Anyone got common cold? Runny nose? Well, you saw me last week. I was this person. Technically speaking, it was a species. Some species, there are 200 different species, literally 200 different species of common cold virus, rhinovirus. When it got in me, I had probably several hundred what's called quasi-species. When virus reproduces, its RNA accumulates mutations, and I have a whole bunch of different viruses in my cell. And then they come out, and some of them will survive, some of them will not, okay? And basically, these 200 species are the versions of the viral genome that are most stable, survivable. Does that make sense? Now, what's the function? So RNA is super error prone. DNA, what's the function of DNA? First and foremost, storage of genetic information. Do we need mistakes in the storage? No, we don't. DNA replication is very error-proof. It's very reliable. There are a lot of proofreading, corrections, and stuff like that. Does that make sense? So, it's your mistakes. But the majority of mistakes are induced by what we call external mutagens. Which two main types of external mutagens do you know? It's much simpler than you think. Huh? No. Mutagens. Things that can cause mutations.
Carcinogens?
Okay. By carcinogens, what do you refer to? Huh? Can you generalize? Starts with C and continues with H. Chemicals. And the second type?
Environmental?
Well, chemicals are environmental. What? Genetics is going to be your replication mistakes. You never watched like a Fallout game? What happened in Fallout, the game?
Radiation?
There you go. Nuclear apocalypse, radiation. So chemicals and radiation of all sorts. Two main mutagens. Okay? Does that make sense? So there are two big types of mutations. One is point mutations. In point mutation, there is a change in a single base. So first type of point mutation is silent mutation. So look at this DNA, which contains GTA codon that encodes amino acid valine. Can you see that? Can you see the GTA that I put in that frame? Encodes valine. If A in the GTA is changed to T, the codon becomes GTT. Turns out that GTT also encodes valine. Are there any changes in the protein? No. So that's it. No phenotype. Mutation is present, but there's no phenotype, therefore silent. Second type of mutation is missense. Missense mutation results in the substitution of one amino acid by another. So here we have amino acid proline encoded by the codon CCC. Okay? If first base C in a CCC codon is replaced by adenine, it will give us ACC, which will change the amino acid sequence from proline to threonine. Does that make sense? Now I'm going to ask a tricky question. Missense mutation. Will it result in the changed protein structure always? Not necessarily. Because if one amino acid is replaced by another that is very structurally similar, does that make sense? Nothing's going to happen. Like in this case, something's going to change because proline is hydrophobic amino acid, threonine is polar amino acid, so something's going to change. But if somehow leucine is replaced by isoleucine, chances are nothing's going to happen. So missense mutation may or may not result in some kind of a phenotype, in some kind of a change in the protein structure and function. Bless you. Are we clear? And finally, the third type is nonsense mutations. In nonsense mutation, the amino acid codon, in this case TAC, here, is replaced with a codon for stop. So this single C is replaced with a G, giving you the stop codon. Does that make sense? So what do you think is going to happen to the protein in case of nonsense mutation?
Totally useless.
Most likely, yeah. It's going to be what we call truncated. So say you have a protein that is 100 amino acids long, and amino acid number 50 is replaced with a stop codon. So now it's not 100, it's just 49. Does that make sense? So sometimes for large proteins, it may lead to some barely functional ones. For instance, the protein cannot perform its enzymatic function, but the signal that directs it into the nucleus may remain. Does that make sense? So cell usually responds to accumulation of dysfunctional proteins. If there are too many of them, it responds by killing itself. If it cannot recycle them all, it just dies. Okay? Which is kind of a basis of mechanisms of actions of some antibacterial drugs. Now that's point mutations. Now you see point mutation. It's one nucleotide becomes another, one way or another. Does that make sense? Now, second big type, frameshift mutations. Now before we talk about frameshift mutations, I want to quickly talk about reading frame. So I'm going to write some absolutely generic sequence of nucleotides. So what's going to be the first codon to get read? AUC, very good. It's going to be the second, third, fourth. So if you look at this, you can see that, and that's, now let me ask you, what this blue sequence that I put there, is that DNA or RNA? RNA. Okay. So Q, uracils, RNA. So you can imagine ribosome.
It's a weird RNA because there's a T.
Oh yeah, my bad. Let's replace it.
It's hybrid.
That happens. You can be incorporated in DNA sometimes. Now, for our purposes, do you see the frame that ribosome uses to read the RNA? Basically puts the frame over the first three, then it moves all the way to the second three. There is no overlap. Does that make sense? You understand what I'm trying to say? It's called a reading frame. Make sense? Now, this was your original DNA right here. Wild type. Okay? On the bottom, to the left. Now, in frameshift mutation, say a couple nucleotides, T and A, are removed. See that? And the entire reading frame immediately shifted. So original reading frame was AGC, GUA, CCC, UAC. Now it's AGC is still there, but now you have GCC, CUA, CUU. So amino acid sequence becomes a complete gibberish. Does that make sense to you? We're good? So frame shift occurs in case of deletion or insertion. Now, in order to get the frame shifted, you must delete or insert 1 or 2 or 4 or 5 or 7 or 8. Does that make sense? Because if you insert 3 nucleotides, what's going to happen? You just add an extra amino acid, right? If you insert 6, you add 2 extra amino acids. 9, you add 3 extra amino acids. Does that make sense? Usually frameshift mutations have an absolutely detrimental effect on the protein structure. It becomes like absolutely dysfunctional. Does that make sense? We're going to take a break, and when we come back, we'll talk